Welcome to this third joint webinar from the PGC and the ISPG. This is the Worldwide Lab webinar on genomics, short read, long read, and blended genomes. We're super happy to have you all here, and we hope you enjoy this first webinar. Please put your questions in the question and answers, and we'll read them at the end. So welcome again. And Niamh, you can start presenting our first presenter.
Well, I'm very pleased to be able to present a colleague of mine, Cathal Ormond, who works here at Trinity College Dublin. Cathal is a postdoctoral research fellow working with the PGC Pedigree sequencing working group. His work focuses on bioinformatics and statistical modeling for family-based rare variant analyses, and he's going to be leading us off talking about short read whole genome sequencing. Cathal.
Great, thank you very much for that introduction, Niamh. I'll just pull up my slides. Hoping that you can see these on the stage, but do let me know if you can't or
Yes.
if there are any issues. Let me get my pointer. Great. So, as Niamh mentioned, my name is Cathal, and my presentation today is going to be focusing on using short read whole genome sequencing to evaluate families with multiplex psychiatric disorders. And I have nothing to disclose. So as Niamh mentioned, I'm a postdoc here working in the PGC Pedigree sequencing group, which is led by Professor Aidan Corvin in Trinity College Dublin. And as this image here shows you, our analysis looks at large multiplex pedigrees with the same or very similar diagnoses. And our goal is to try and identify shared genomic factors that might be increasing individual liability for the common or similar diagnoses. And by using short read whole genome sequencing, we can evaluate quite a wide spectrum of genomic variants, including single nucleotide variants, indels, and copy number variants. So my presentation really is going to focus on the various different kinds of analyses that we can implement by using short read sequencing and just various different family studies that we have looked at in the past. So the first cohort I'm going to describe is a collection of schizophrenia pedigrees that were recruited by our collaborator, Professor Bill Byerly in UCSF. So here are 7 of the pedigrees for whom we had generated sequencing data on about 55 individuals. So those marked in black have a diagnosis of schizophrenia, and those in the green dot are those with sequence data. So our pedigree structures are a little bit varied, and we have at least 4 or 5 individuals per pedigree who are affected and sequenced. But I guess our rationale here is that there is likely going to be a segregating variant or maybe multiple variants that are implicated in schizophrenia. So our first pass analysis that we typically implement is to look at protein coding regions. So in this analysis, we started looking at SNVs and indels, and we wanted to find family private variants that had perfect or maybe slightly less than perfect segregation with schizophrenia that were also in some ways damaging. And we appealed to the schema rare variant exome analysis, which showed enrichment of ultra-rare protein truncating or highly deleterious missense variants into constrained genes. So we started off with about 12 million variants genome-wide, and when we focused on protein coding variants and then applied our filters, we ended up with 3 variants that had reduced segregation patterns with schizophrenia. And again, this was pretty expected since schizophrenia even in complex trait even in family-based studies. So these 3 variants were all predicted to be damaging more or less by other deleteriousness scores, but the one that stood out to us was in this gene ATP2B2. So here I'm showing you the family with the rare variant carriers. The plus underneath indicates one of the rare variant carriers. So we have 4 out of the 5 schizophrenia cases were accounted for, and then the variant was found to be absent from our control members, and it was absent from the other 6 pedigrees in the cohort. So when we published, we thought this gene was interesting because it's involved in intracellular calcium homeostasis. It's found to be brain expressed, or at least across multiple brain tissues in GTEx. And there was a common variant meta-analysis that identified this as a fine map gene when looking at common variants between schizophrenia and autism. But since we published, this has come up in the bipolar exome BIPEC study, which I won't talk about at all because I appreciate that is at the basis of Cal's talk. But we thought this was an interesting gene because it seems to show converging evidence from certainly our small family study with other disorders that are quite related to schizophrenia. The next analysis we were interested in from these data was to look at copy number variance. So calling CNVs from short read data is certainly possible. At times it's a little bit more of an art form than a defined science. But the approach that we fixed on was to look at a consensus approach to take 4 CNV calling software tools that have reasonable accuracy and then combine them by taking CNVs found by at least 2 out of the 4 callers. And this is a method we had published on a couple of years ago. So again, after calling CNVs from our short read data, we wanted to find those that segregated with CNV that were with schizophrenia, should say, that were particularly rare and in some ways damaging. And following our filtering, exactly 1 CNV was left following manual review. And this was a 3 kilobase pair deletion that overlapped an intron-exon boundary in the gene PIT or M1. And you can see in the pedigree diagram, it did segregate perfectly with schizophrenia in this small family. So in ClinVar, clinically or pathogenic variants in this gene have been reported for a complex neurological phenotype that involves seizures, ataxia, and developmental delay. But the same pathogenic variants had also been found in family-based studies where adult carriers also showed symptoms of psychosis and even schizophrenia. So again, we think this is an interesting gene, and it may go some way to explain why in our family the deletion carriers may have a diagnosis of schizophrenia. So this is our standard, as I might call it, bread and butter style analysis by looking at SNVs, indels, and CNVs that overlap protein coding regions. The next analysis that I was involved in is a little bit of a different study. So we looked at a large pedigree, or maybe multiple pedigrees, it's better to describe them at, that had individuals diagnosed with Tourette syndrome. So these pedigrees were collected by Professor Cara Matthews and were recruited from Costa Rica. And I should point out that a lot of the primary analysis was done by our lovely chair, Niamh, here. I was involved partnering with it, so it's why I'm describing it. So originally, these individuals were ascertained as trios for Tourette syndrome. So you can see that the 9 individuals I selected here from as a subset of our pedigree all have a diagnosis of Tourette syndrome, and their parents were not found to have a diagnosis either. But by using genealogical records, this pedigree structure could then be reconstructed. So we can see that actually some of our trios are quite distantly related to each other. Now, this is not a typical family study as we might think about it, because when we usually look for variants shared across individuals with the same diagnosis, that assumes that they might share a common ancestor that might be quite close in the pedigree structure. But of course, given that we have these very distant cousins to each other, it's quite possible that any shared rare variants might have entered the family through marrying individuals. And those with sharp eyes will also note that at the top of the pedigree, there actually are multiple pairs of individuals who are ancestors of these 9 individuals out of the total 19 we had. So it becomes quite challenging to try and think about this as a standard family analysis. So instead, the analysis approach that was selected was to look at identity by descent segments. So taking common variants or a subset of the common variants from the whole genome sequence data, these were phased relative to the 1,000 genomes admixed American subcluster, and then the phased chromosomes were used to generate long haplotypes on an individual basis. So long segments of the genome that appear that they have been inherited from one individual somewhere back in that individual's lineage. And then by looking at individuals in the pedigree structure who are known to be related to each other, we can identify these multiple IBD segments, which are very likely identified or are inherited identical by descent. And then to kind of strengthen the analysis, we can look at individuals who are a control population taken from the same region as our pedigree cases and remove any haplotypes that are found in this control population. So to really strengthen the idea that these haplotypes have occurred from a common ancestor, even if it's many generations back, and that really haven't been inherited from individuals coming in later in the pedigree who may have no connection to the diagnosis of Tourette syndrome. Then turning back to our sequencing data, we can fine map the rare variants by examining the individuals who carry the specific haplotype and by examining the alternate alleles present in those. So in this way, we can backfill in all of these rare variants that are carried by individuals who share a haplotype identical by descent, where those variants are also absent for everyone else in the cohort. So this kind of haplotype identity by descent analysis stands instead of our standard or straightforward segregation analysis. So following this, we looked at rare variants that were also predicted to be in some way damaging, and we found 4 rare variants in 4 genes. Now, none of the genes had an extremely strong connection or had shown a strong previous evidence for connection with Tourette syndrome, although two of the genes in particular, RAPGEF1 and ERBB4, did show a very strong connection to neurodevelopment. So we think that they are quite good candidates for Tourette syndrome for the haplotype carriers. But one thing I'd like to point out is that we didn't just focus on protein coding variants. We did identify 2 missense variants, but the analysis strategy was expanded to include intronic variants as well. And you can see that 2 of the haplotype rare variants were also in introns. So I think this is kind of a great study that really shows the depth and diversity of what can be done with short-read whole genome sequencing data, in that you can take common variants genome-wide and use them to construct haplotypes, which can then be used to identify rare variants, either coding or non-coding, that might have be strong candidates for being responsible for Tourette syndrome in some of these individuals. So then the last cohort I want to chat briefly about is some of our more recent analysis. So these are 2 large OCD pedigrees, again, that were recruited by Professor Carol Matthews in the United States. This work is still under review, but we do have a meta-archive paper, so I'm going to talk about some of the results that are in that preprint. So we selected 15 individuals from this pedigree, marked in green again, who have a diagnosis of OCD. So it is quite a large pedigree, and again, we have one common founder here, so it is plausible that we might find a rare variant that is inherited or carried at least by several individuals in our who we'd sequenced. So we had implemented our Bayesian prioritization approach called BICEP to try and prioritize our protein coding variants as our first pass analysis. There were no rare CNVs or indels that had good segregation in this pedigree, but we did find one rare missense variant which had reasonable segregation with OCD found in the gene NPY5OR. You'll note that it isn't carried by all 15 of the affected sequenced individuals, but it was found in the majority of 10 out of 15 cases. And this gene hadn't come up in previous common or rare variant studies for OCD, but it had been implicated in GWAS for panic and addiction disorders. We think it might be a reasonable candidate for this pedigree. The second pedigree in this cohort was a little bit trickier because, again, we have 2 unrelated founders from which all of our pedigree cases seem to be descended. We had selected 10 individuals in this pedigree, most of whom had a diagnosis of OCD for sequencing. So since we have 2 separate unrelated branches of the family, we did analyze them in isolation, and it turns out that some of the more interesting results did come from the smaller leftmost branch. So our Bayesian prioritization approach identified 2 variants that achieved the maximum achievable segregation score. The first was a rare missense variant in MAPK8IP3, which doesn't have a very strong connection to OCD, but the second one was a rare deletion, a copy number variant in DLGAP1, again, which has strong segregation with OCD. Now, this was an almost 1 megabase pair deletion of the gene. It overlaps the first 3 exons in the untranslated region, but DLGAP1, also from the SAPAP gene family, has come up multiple times over in the literature as a potential risk gene for OCD. So we think this is a reasonably strong candidate for why the individuals in this smaller sub-branch of the family might have increased OCD. So I suppose up until now, we've really just focused on rare variants because that is what we do a majority of our work in. But I did want to highlight that something that we can look at quite straightforwardly from whole genome data is polygenic scoring. So for our 25 individuals from across our 2 pedigrees, we generated polygenic scores, accounting for the fact that we do have extremely highly related individuals here. And we compared them against a background distribution which was taken from the European subset of the 1000 Genomes Project. So this black bell curve is the polygenic distribution from our unrelated population controls, if you will. And we could then project our pedigree members onto this distribution. Now, again, we don't have the power to say anything very definitive about the study, so I don't want to overinterpret this. But we certainly didn't observe any immediately obvious strong trends from our polygenic distribution. So it does seem that the rare variants are the most likely candidates for why these individuals may have OCD across these 2 pedigrees. And then the last thing I want to talk about just very briefly is, I suppose we've seen from the cross-disorder group and from more recent rare variant sequencing studies that OCD and chronic tic disorders like Tourette syndrome have a shared genetic biology, and that we certainly would expect to find some commonality. So we wanted to investigate if the risk genes that had come up in our large Tourette's family showed any kind of commonality or interaction with the genes that had come up from our OCD study. So here is a string protein-protein interaction network that we generated looking at the 4 genes that were impacted by rare variants in our Tourette study and 4 genes impacted by rare variants in our OCD study. And we focused on the highest confidence interactions to really kind of not just be searching through a lot of noise. And you can see in the center that we did observe that there was a cluster where 2 of our genes from our Tourette study did find interaction with 2 of our genes from our OCD. So this does show us, this does again support the evidence that certainly from rare variant studies, we can find an interaction in terms of the genetic etiology of OCD and Tourette syndrome. And I think what we quite liked about this was that some of these rare variants were missense, some were introns, one was a copy number variant. So it really just does highlight the diversity of genetic variation that's available and how that can help us piece together some of the biology of these quite related traits. So I think I'm at time now, so I'll leave it there. I'd just like to thank very much our research group and all of our collaborators and PIs and researchers who have really helped bring all of these projects to fruition and to completion. So thank you very much, everybody.
Thank you so much, Cathal.
Thank you so much, Cathal. That was a great presentation. And now we move on to the second presentation, and I want to introduce Natasha Berthold from the University of North Carolina at Chapel Hill. She's a postdoctoral research fellow in the Anton Lab at the University of North Carolina at Chapel Hill, with a PhD in psychiatric genetics, focusing on eating disorders from the University of Western Australia. Her current work focuses on understanding the neuronal circuitry, underpinning neurodevelopmental and psychiatric conditions, and elucidating the genetic and cellular mechanisms involved. Thank you so much for accepting this participation, and you're welcome to share your slides.
Okay, perfect. Can everyone let me know if they can see that all okay?
Yes.
And again, just like
Perfect.
Cathal said, if at any point it drops out, just let me know. Okay, so yes, my name's Natasha. I am a very, 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 very brand new postdoc. And so it's a real privilege to talk to you today about long-read sequencing. I'm going to take a little bit more of a broad view than Cathal did and talk a little bit more about the concept and the application, potential application of long-read sequencing to psychiatric genetics as a whole, using a study that I did as part of my PhD project at the end as kind of a use case example. So to kick us off, so the challenge of psychiatric genetic architecture, I mean, I imagine most of you here today have worked to some extent with psychiatric genetics. So I think, it's fairly well known by now that there are several challenges when it comes to characterizing the genetic architecture underpinning psychiatric conditions. First of all, phenotypes. Phenotypes are constantly a big question. My specialty is eating disorders. So, diagnoses are not rooted in biology. There's always the question of, are we even looking at the same thing? One thing that I was thinking about as Cathal was talking was, is it a collection of like all these very rare biological conditions that end up presenting kind of similarly, like lots of different paths to a similar outcome? There are so many limitations to the DSM-5 and diagnostic questionnaires. Part of these large-scale GWAS studies, which are incredibly important for genetic discovery, do really, really, really simplify down these very, very complex phenotypes. You will get 2 people with anorexia nervosa, and even if they meet the diagnostic criteria, their experiences are vastly different. And I think those types of things are really, really important as we start to get through this first kind of era of GWAS discovery and start to probe more of these like complex underlying questions of, again, what Cathal was talking about was like, okay, there's these risk genes that are popping up across all these different conditions, but how are they working for these different phenotypes? So on that note, another main challenge with psychiatric genetics is the complex biology. So as you heard, my postdoc is in more like mapping the neuronal circuitry in neurodevelopmental and neuropsychiatric disorders. So taking a bit more of a functional approach. And, it never ceases to blow me away, just the structural and functional complexity of the brain, both inside and outside of psychiatric disorders. Just looking at how the brain functions in a quote unquote normal manner. How do we map? How do we go from the genetics to that incredible complexity at not just like the cellular level, but even within the cell and subcellular localization of proteins, and how a protein expressed in the soma of a pyramidal neuron can be different to the same protein expressed in a dendrite of a hippocampal neuron or something like that. So all of that to say, like, the journey from DNA sequence to observable phenotype is incredibly long and complex, and we can't just rely on the much more traditional method of going straight from genetic variant to phenotype. So finally, another main challenge in understanding psychiatric genetics that I want to talk about before I get into my long-read sequencing story. So we all know by now, genetic architecture in psychiatric conditions is polygenic. It's pleiotropic, it's epistatic. There are diverse variant types. Again, Cahill talked about, single nucleotide variants, copy number variants. Then there's transposable elements. There's tandem repeats. You can have short indels, or you find these insertions and deletions or complex rearrangements that can span up to kilobases in length. So all of this requires a really multi-dimensional approach to understand how we get from DNA sequence to biology to final phenotype. So now on to the fun part, the long-read sequencing. So what is long-read sequencing? Long-read produces long reads up to ultra long reads. So this is up to hundreds of kilobases in length. Typically, a long read will be 10 up in the tens of thousands of base pairs. There are two key players in the long read game at the moment. That is Oxford Nanopore Tech and PacBio. Now, both of these offer long read sequencing options. There are some slight differences that come with them. So the methods of sequencing and the raw data output differ slightly between ONT and PacBio. So for example, ONT uses the famous motor protein pore based sequencing known as nanopore sequencing. And this was the type of long-read sequencing that I did during my PhD. So I will be focusing more on nanopore in this talk than PacBio, but I thought it was pertinent to mention PacBio. And interestingly, a lot of people are now combining both ONT and PacBio to kind of bring together their strengths and cancel out the limitations of each. So PacBio, on the other hand, doesn't use the protein pore. It uses a hairpin adapter-based sequencing method, which they call SMART technology. So ONT tends to produce much longer reads, but they tend to have a lower base pair accuracy, whereas the PacBio Hi-Fi long read has shorter reads, but at a higher accuracy. Again, you can start to see where people are thinking of using both of these. One of my favorite facts is that the ONT raw data is called squiggle data, because it quite literally, the way ONT nanopore sequencing works is you have a plasma membrane with little motor protein pores in it and a ionic solution. You run charge through the solution, and as that charge is run through the solution, it will force the DNA to be ratcheted, to unwind and be ratcheted through each of these motor protein pores. And as each base pair goes through, it causes a change in the electrical signal, which is picked up by a sensor membrane underneath and read out in real time to your user interface. And so it's literally read out in this little squiggle signature, which is then converted either, in real time programmatic base pair calling, or you can, as I'll talk about, go and do your own base pair calling in bioinformatics. Whereas on the other hand, the PacBio raw data is read out as BAM files. So the final key thing that I'll be talking about when it comes to long-read sequencing is the bioinformatic analysis aspect of long-read. And this is a really key point. So obviously the chemical and technological aspects of long-read sequencing are super useful and are constantly being developed at the moment. But in terms of really optimizing long-read sequencing for psychiatric genetics, a real rate limiting step is this bioinformatic analysis component. So, pros and cons of long-read sequencing. So I think it's fairly evident that the long to ultra long read length is a vast improvement over, well, hi-fi, short hi-fi is great. The long to ultra long is an amazingly powerful tool for mapping breakpoints. You know, if you have reads, so very similarly to short read, you will get your alignments, you'll get your reads, and then when you're doing bioinformatic analysis, it will stack the reads over each other and come up with the best likely sequence in that region against a reference sequence, or if it's doing de novo. So you also get nucleotide resolution, which I think is a really, really powerful point, particularly when we're starting to look at psychiatric conditions. And again, to refer back to Cahill's talk and talking about how you'll get, you know, these polygenic scores, and you'll get these pleiotropic signals that are indicating that, you know, one gene or one risk region is implicated in several different conditions, and then you're wondering, okay, well, is it doing the same thing in all these conditions? Is it that it's doing something different in these conditions? If it is doing something different, is it there's this one mutation or this one variation, and it's when it's exposed to like an external influence that it makes it produce a particular output? Or is it that there are different variations, and if you get a particular variation in that region, or a particular constellation of variations in that region, that you get result A as opposed to result B? So I think the really cool thing about nucleotide level resolution is that you can map, you know, copy number variants or large insertions and deletions, and you can actually like pinpoint particular within mutation, within variant variation that can explain, you know, dosage effects. It can explain like subtle differences between, again, like psychiatric conditions are not these binary case control diagnoses as much as, you know, that's been useful. Psychiatric conditions occur across a spectrum. So we have to start thinking about them as more of these continuous variables rather than these case control binary variables. So I think like the power of the nucleotide resolution is going to be really, really useful for that. The other really, really cool thing about long-read sequencing is that it captures methylation without any extra steps. Both PacBio and ONT do this. So you don't need to do any bisulfate sequencing. There are absolutely no extra steps required when you're doing your prep, whether you're doing native sequencing, whether you're doing sequencing by ligands, whether you're multiplexing, because long-read sequencing also offers multiplexing. You will get the methylation data within your raw data. And I think, you know, that's really, it's a very powerful additional aspect, particularly when you consider the fact that you can get both DNA and RNA long-read sequencing. The final point that I want to make as a pro is that the raw data longevity compared to array genotypes is better. As I was talking about, you know, with the long-read sequencing, particularly with ONT's squiggle data, I have to smile every time I say that. But, you know, you can do a long-read sequencing experiment, say in 2022, and then you're able to, you don't necessarily have to, when you do that, the raw data is not necessarily aligned to a reference genome. So you can, as you know, reference genomes update, as our base calling software improves, ONT is releasing updates all the time. It's fairly easy to go through and redo your base calling analysis. Whereas, one of the real issues when it has come to array genotyping is that the data tends to decay faster. Arrays that were, or array genotypes that were done 20 years ago, not quite obsolete, but they're certainly nowhere near as long-lasting as the long-read sequencing data is. So on that note, I do have to talk about some cons to long-read sequencing. So it is very cost prohibitive, unfortunately, which is why at the moment, studies do tend to focus on pre-identified variants that they want to map, or, pedigree sequencing, ultra rare conditions, that kind of thing. However, the costs are significantly decreasing, as we've seen time and time again within the genomic technology space. The downside of long-read sequencing in comparison to something like genotype arrays is that while there are multiplexing capabilities, there is still a much lower throughput compared to the array genotypes. Even if you had the money to sequence 50,000 people with long-read sequencing, can you tell me who your funder is? And that would also take technically a long time in the wet lab. And it would also be bioinformatically incredibly heavy process. On the note of bioinformatics, my final point is that it is still a little bit of a Wild West. And this does make sense. Like, ONT only really started releasing their first kind of trial kits in, I believe it was 2007, thereabouts, in the early sort of 2010s. And so it makes sense, right, that we're still kind of fumbling our way through figuring out how do we actually analyze this data. So the post-GWAS long game, how does long-read sequencing fit into psychiatric genetics? I have 4 key points of how long-read sequencing fits into psychiatric genetics. So the strengths of long-read are really, really, really well suited to the nature of psychiatric genetics. It captures the genetic complexity that mirrors the phenotypic complexity. Again, remember what I was talking about with the way that we have incredibly complex phenotypes that we need to start thinking about in more of a continuous manner than case control binary. And so this is the power of the long read. Again, could it be that there's actually this collection of biologically quite unique conditions that are just manifesting in a similar way? This is where the pangenomes come into it. So pangenomes are made possible now by long-read sequencing. It's basically where you do telomere to telomere sequencing of someone's genome, and you, in the analysis, instead of aligning to a traditional linear sequence, you basically construct multiple different paths that are based on a number of, given parameters or accepted rules about how the genome works. And it will produce, go and have a look at pangenome graphs. I strongly recommend it because they're fascinating to look at. So that captures the level of structural variation within an individual. Again, so the second point is that structural variation not easily captured in GWAS, and poorly resolved regions of the genome are much more easily resolved when we're looking at long-read sequencing. So as I said earlier, it spans breakpoints, particularly for things like tandem repeats and complex variants. It's really, really hard to characterize tandem repeats with array genotypes. When you're using imputation, it's highly unlikely that that's going to actually reflect the true allele. Whereas with short-read sequencing, the problem with short-read sequencing is that it also struggles to map against these tandem repeats, because particularly when the tandem repeats, exceed kind of 100 base pairs, with short reads, you'll get these multiple reads that are just that tandem repeat, that you can't properly judge at what point the breakpoint occurs and how long that allele is. And we're seeing, with complex traits, we're seeing that allele length plays a real role in like dosage effect. So on that note, you can also do intravariant variation. So again, what I was talking about, some of the really cool work that's coming out of Melissa Gimrick's lab, I'll drop that name. They're looking at how, like, within tandem repeats, you may have a perfect tandem repeat, and then you may have a tandem repeat that has, you know, a couple of point mutations within it. And on the surface of it, you might class those two things as just the same tandem repeat. However, they're starting to find, using long-read sequencing, that there's actually differentiation between, you know, these alleles. My second 2 points as to how long-read sequencing applies to psychiatric genetics is that I think it will be incredibly helpful in resolving the similarities and differences of psychiatric genetics. So again, going back to that point that I made about how this ongoing question of how can these genes be polygenic and pleiotropic for different psychiatric traits? You know, are they doing the same thing? Is it when you have a cluster of different variations? This is where the long read is going to be super powerful for characterizing intrapersonal variation and interpersonal variation, and starting to, like, pass what is functional variation for a particular condition that you're looking at, and starting to, you know, help map perhaps these more, like, distinct biological groups that end up in these broader phenotypes. And finally, you know, long-read sequencing is very readily and very easily able to be integrated with functional data for much more comprehensive biological picture. I haven't done it myself, but I will make a quick mention of spatial transcriptomics with ONT, which is absolutely amazing RNA sequencing of, like, particular parts of the cell, so you can understand where in real time certain genes are being expressed within a cell. So to wrap up, the last thing that I will talk about is the use case that I used in my PhD of long-read sequencing for anorexia nervosa. So I'm aware not everyone here is an eating disorder researcher. So very, very quickly, what is anorexia nervosa? It's an incredibly complex condition. It's highly heritable. Well, you know, the heritability, twin studies have shown that it can be up to 75% heritability. And in to date, the most significant GWAS done, 2019 by Hannah Watson et al. They discovered a SNP heritability of 11 to 17%. Keep your eyes peeled for the PGC eating disorders most recent GWAS, meta GWAS, which will be coming out very shortly. According to the DSM-5, anorexia is characterized by 3 key criteria: nutrient intake restriction, a drastic change in weight and shape, which is unfortunately still measured by BMI, and a pathological fear of weight shape change. Just in the interests of time, I will quickly skip over describing too much of the biological environmental prevalence factors, but, you know, there's been a significant amount of neurobiological work that's been done in anorexia, and it is very, very much not just a social disorder as it has historically been considered. So in my PhD, that went too fast. There we go. In my PhD, we decided to do a exploratory analysis of these 10 case control samples down at the University of Otago in New Zealand in Martin Kennedy's lab. So we wanted to establish and apply this targeted nanopore sequencing method. So one thing that I didn't mention,
Natasha,
just aware of time.
I'm so sorry, just to say, just be aware of time. Yeah.
Okay, yeah. I will very quickly run through. So take a quick look at the pipeline that we applied. So basically we targeted the specific 8 regions. This just meant that we would have better coverage over those regions as opposed to doing whole genome sequencing. What we found was that these regions, there was so much variation that just was not captured in databases. Unfortunately, you know, we lacked statistical power and we only had participants of European ancestry and female sex, but it really, really highlighted just the power of long-read sequencing to characterize these risk regions. So to close out my key takeaways, I have 4 key takeaways, is that the bioinformatic analysis is a rate-limiting step, but it is surmountable, and particularly as we continue to innovate and accumulate data together, plug for our group science. The power of the pangenome assemblies for better understanding individual functional versus non-functional genetic variation. So genetic variation that's just a private mutation that doesn't mean much, and genetic variation that contributes to a particular phenotype. The powerful information to integrate, it's powerful information to integrate with functional analyses for a much more full biological picture. And finally, integrating it with phenotypic and environmental data to really get that more complex, multifaceted view of psychiatric conditions. So thank you so much for letting me speak to you all today, and I will pass it back to Niamh.
Thank you
Thank you,
so
Natasha.
much.
Great presentation now.
So in the interest of time, I'm going to move straight on, and I have the pleasure to introduce Cathal, who is at the Broad Institute of MIT and Harvard. Cal is a postdoctoral research fellow at the Broad Institute and Massachusetts General Hospital, whose work focuses on developing genetic and multi-omic approaches to understanding psychiatric disorders. He is currently the lead analyst for the Bipolar Exome Consortium, BIPEX, and contributes to large-scale sequencing studies of bipolar disorder and schizophrenia.
All right, thank you so much for the introduction. Let me know if the slides are okay. I'm doing presenter mode. Perfect.
Yes.
All right, so I'm excited today to talk about rare variant discovery and bipolar disorder on behalf of the BIPEX Consortium. And I always like to kind of highlight this review paper, which essentially contextualizes discovery of genetic variation across depression, schizophrenia, and bipolar disorder. And really what we can kind of see is that for the common variation across these three disorders, there's a decent saturation in terms of discovery, but then when we pivot over to rare variant discovery, it's fairly non-existent in bipolar disorder and depression relative to schizophrenia, where you have a lot of discovery in terms of protein truncating variation and then also copy number variation. And so this segs really nicely to the BIPEX Consortium, or the Bipolar Exome Consortium. In the first iteration, where we had 14,000 cases and 14,000 controls, but we ended up having no exome-wide significant hits. However, ACAP11 did emerge as a definitive risk gene when combined with schizophrenia data. And I think a lot of this can be chalked up to the fact that we're just fairly underpowered to detect genes despite this large sample size. And so we've launched several large-scale initiatives, like the populations underrepresented in mental illness association studies, where the goal is to build these large-scale biobanks across Latin America and Africa, where we have around 40,000 individuals with schizophrenia, 40,000 with bipolar disorder, and 40,000 matching controls. To highlight a region within Colombia that is super important in understanding why we care about of having these global efforts. For instance, the Paisa region has a 8% prevalence of bipolar disorder relative to the 1 to 2% in North America. So again, these unique genetic architectures that may enable us for more powered discovery. We also have the Asian Bipolar Genetics Network, or ABIGNet, and the idea here is that we sequence across Asia around 30,000 cases or so and 30,000 controls. But because we're looking at these diverse populations, we had to develop a technology called the blended genome exome. And the idea here is that we have this low coverage genome around 4x to capture this common variation that is not well detected through canonical microarrays, which tend to have this Eurocentric bias. And then you have this deeper whole exome around 30x, which allows us to capture this rare variation within the codons that I'll be talking about today. For BIPEX 2.0, we've vastly increased the recruitment.
I'm so sorry. Is your screen dark? We've had some comments that it's quite gray.
I don't think so, but maybe I'll just get out of presenter mode.
Yeah, it's brighter. It's brighter there. Yeah, for some reason it goes dark. It's like it's grayed out a little bit. That's much better. That's it. Yeah.
Okay.
Perfect.
I guess this was my
Thank you.
presenter mode, so I won't use it. Okay, so for BIPEX, the recruitment, we vastly increased it. So now we're around 65,000 cases and 170,000 controls. And again, this kind of recruitment across the entire world. For the sake of time, I'm not going to get too much into it. But I do want to take a second to highlight how much the ancestral diversity has increased for BIPEX. So I put the numbers on the bottom here. But we can really see that not only is this a very global effort across 22 countries in the world, but genetic diversity has also been vastly increased in the BIPEX study. And so what we want to look next is at these ultra-rare variants within different gene sets to kind of serve as a positive control in understanding exactly whether our data makes sense. So we have these gene sets under high purifying selection, so constrained genes, a neurodevelopmental disorder gene set, autism, and then schizophrenia. And the idea here is that we're just assessing across these different variant types, so protein truncating variation, which is predicted loss of function, and damaging missense. And then we see that across all four of these gene sets, we have this significant enrichment of these variants in bipolar disorder compared to controls. However, for synonymous variation, where there isn't any change in the protein, we end up not seeing any signal there. So what we did next is we want to combine the protein truncating variants and the damaging missense to do a gene level association test. And so earlier we had 0 genes that we found for the first iteration of BIPEX, but now we have 13 Bonferroni significant genes, and with an additional 20 that are FDR significant at 5%. I want to take a second to kind of appreciate how large the effect sizes are for these. So you can imagine you have a baseline risk of bipolar disorder at 1%. And if you were to carry a loss of function variant in ACAP11, which has an odds ratio of around 10, then this increases your risk of bipolar disorder to 9 to 10%. So these really, really large effect sizes, with also several of them being strongly associated with other phenotypes like autism, so ATP2B2 and KDM5B, and then many also associated with schizophrenia like SP4 and ACAP11. And so when we were looking at the data, we next wanted to kind of parse out, well, what genes have a stronger missense signal or what genes are purely loss of function. And so for a gene like ACAP11, we see it's purely driven by loss of function variation only, which is really useful from a functional model perspective of exactly trying to understand what we may want to model. And then you have some genes like ATP2B2, CUL1, which are actually driven a bit more strongly by missense signal instead of loss of function. And so this serves as a really nice motivation for us to assess this 3D missense localization across a protein, and that can we use this to create mechanistic hypotheses that we may want to follow up on functionally. And so we developed a 3D protein neighborhood test, and what that really just means is that we're able to kind of scan through the protein, these AlphaFold 3 proteins that we pull, and then look at these 3D angstrom scans across the entire protein and kind of ask, well, within those regions that we're looking at, do you see an increase of these ultra rare missense variants in bipolar disorder compared to controls? And so we did this for the significant genes of BIPEX, excluding ACAP11 because it was purely driven by loss of function. And we end up finding 2 signals, so one for ATP2B2, and then 1 signal for DOP1A in that protein for that 3D missense signal. So for ATP2B2, it's the strongest association for that missense signal is actually localized to the calcium binding site. And so this, from a mechanistic perspective, it's fairly understandable. There's probably the damaging missense is perturbing that potential binding of calcium. But then for DOP1A, we actually found no obvious domain or cofactor binding site where the red indicates the strongest signal for missense in that 3D protein. And so this kind of led us to wonder, well, what can we do next to kind of interrogate exactly what is happening with DOP1A? And so we pulled all these protein interactors with DOP1A from string, and there's maybe 20 or so, so not too many. And then we fed this into AlphaFold 3, and the idea is we're trying to identify these high confidence predictions for what proteins actually interact based on AlphaFold. And so earlier I highlighted these 13 bond-friendly significant genes, DOP1A. There's also ATP9A, and I serve this as a reminder because what we see on the next slide is that at that region with that increased missense signal, AlphaFold 3 is predicting that ATP9A, which is another BIPEX gene, seems to interact or dock there. And, I just want to take a second to kind of really appreciate, we put in 20 interacting proteins. There's only 13 genes that are we find in BIPEX, and we come up with this type of potential mechanistic hypothesis where these two BIPEX genes are potentially interacting at this localized 3D missense signal. And so there's not too much on the biology of what these two proteins are doing, but kind of high level sense for DOP1A, it seems like it's involved with the Golgi membrane trafficking. And then ATP9A is kind of really focused on the formation of these types of vesicles for the Golgi. And so potentially there may be some mechanistic thing going on for the Golgi that we may want to further interrogate a bit more, but it will just require more functional studies for us to really understand. And so just a couple conclusions. One, we consistently see that rare coding variants have the largest effect. These genetic risk factors do not respect the diagnostic boundaries of the DSM-5. So we see ATP2B2 being associated across several phenotypes like autism, schizophrenia, and bipolar disorder. We're able to leverage spatially structured missense, which points towards these mechanistic hypotheses that we may be able to follow up on functionally. And as we scale up sample size, we're confident that there will be more genes to come and enable our discovery in bipolar disorder. And so I want to thank all the people on this slide. I'm sure I'm missing some names, but I'm happy to take any questions. And of course, all the participants that enable the type of research we do.
Thank you so much, Cal. Can I also congratulate you formally on the Nature Genetics paper for the BIPEX study? I mean, it's wonderful to have that out now, and I'm sure that a lot of attendees are going to go away and be reading up on it now. Sorry, the blended exome genome paper. Yeah.
Yeah. So the BG method we recently published, but for BIPEX, we're hoping to have that preprint out sometime this summer, and then update.
Very exciting. We're very much looking forward to that. Thank you to the three presenters for a really fantastic whistle-stop tour of the different types of genomics projects that are happening both within our field of psychiatric genetics, but also the potential for how you can use different cohorts and different study types to try and answer different questions. And there have been a lot of parallels, I think, across the different talks in terms of things that are coming through about, phenotypes being different, but maybe the same genetic variants are cropping up. ATP2B2 being both a BIPEX gene, but also coming up in the family study that Cathal was presenting shows that we're transcending boundaries in the disorders we're studying and the types of analyses we're achieving. Do we have questions coming up in the panel at this point?
We do.
We do. Brilliant.
We do. There's one that was answered. I don't know, should we go just to the open ones?
I think, do you want to go straight ahead there, Jetty? And
I'll read the first one from Joanna Jimenez.
Yeah.
Have you tried another model like ESM-Fold2 or Proteinics for another protein configuration modeling?
Yeah, that's good. I haven't yet, but I can definitely add that to the list. I guess we've just been so enamored with the progress of AlphaFold 3 that hasn't even, we didn't even think about the idea of using other predictions.
Um, for Cathal, we've got a question. I think it's for Cathal because it's about family. So, apart from calculating polygenic risk scores and comparing familial cases, familial controls, and population controls, are there other approaches to integrate common and rare variants for analyses across families and across case control cohorts? For example, are these methods such as familial genetic risk scores or other integrative frameworks that would be appropriate?
Ooh, excellent question. So yeah, I mean, that's something we're kind of, I think, struggling with a little bit at the moment, that a lot of the research that I've read just really looks at a broad level, even in case control cohorts, of polygenic score versus maybe rare variant burden, which I think is totally valid. And I suppose we've seen there's an inverse relationship to some extent across, the likes of schizophrenia and autism, maybe less so for other traits. I'm aware that there are family genetic risk scores that have been developed. I think it's by Ken Kendler and some of his team that try to aggregate in some sense, without even looking at genetics, just the family risk for a specific disorder by looking at large pedigrees and by looking at the recurrence of various different disorders across multiple generations. And that can be in some way predictive of rare variant burden. So I suppose it's something you can do without needing to generate lots of data. But I think it is a little bit of an open space, that it is kind of challenging to try and model all these very disparate effects, because I suppose even rare variant burden itself is something quite difficult to try and tease apart. If we have lots of protein truncating variants, I suppose they're very straightforward to interpret, but it gets kind of quite a bit more challenging where we have these more subtle effects. And certainly for families, I suppose we need lots of large families with lots of large sequence data, which is, I suppose, is quite a limiting factor.
So I think I can see other questions on the chat, and
I think at the moment
questions.
we've got all the questions. Sorry, I think we've got all the questions from the chat, but please add more. Whilst we're waiting for more questions, one thing that I thought came through from this talk was how much we're stepping beyond GWAS with this webinar today, and the scope of what can be achievable, particularly looking at the trans ancestry and more global equity in genomics research from using these different technologies compared to using GWAS. You all have different benefits and issues with the different technologies. Do you each of you want to give like your one favorite thing about the technology that you were presenting today? I think that would be a really nice way to kind of show people the showcase of why you would choose the short read or long read or blended exome genome for their particular study that they want to do. Starting with Cathal.
Sure. I think I kind of said it in my presentation that there is just such a wide variety of variation that you can look at pretty straightforwardly. And I appreciate when we say short read whole genome, that doesn't cover the entirety of the genome, but looking at the vast majority of protein coding and non-coding, getting good resolution on structural variants while only needing 30x coverage and not having to break the bank with large cohorts. Like, I suppose since we have such small families, you can do quite a lot with 5 to 10 individuals and with short read data, because it really just does give you such a wealth of access of genetic variation.
Natasha.
Firstly, the nanopore sequencing technology is just really, really cool. But from a personal perspective, I also really like it because of its ability to map variation and like the resolution that you get of genomic architecture across the human genome. I mean, again, Cathal said like it's expensive, but I do think that the insight that we can get from it will be incredibly powerful, particularly as we start to branch out into different types of variation.
Excellent. Cal.
Yeah, so I think for BGE, what is just really nice is the scalability of how we can do it. So it's less than $100 a sample, which is roughly the same cost as just a regular exome now. But you also get all the common variation in an unbiased way in a single data set, or like it all comes in the same like one cram file. And so it also mitigates these kind of biases potentially from having separate runs, like a microarray separately versus like an exome. So
great. So I'll read the next question. Have people done research comparing the biological conclusions of SNPs to gene mapping methods, specifically with psychiatric disorders like AUD and bipolar disorder? So I guess that's
Anybody who wants
maybe,
to answer.
yeah.
I
I
know
think that
for
can go to anyone.
the latest PGC bipolar GWAS, they essentially took a consensus approach for identifying what was considered to be their, I think it was like, I don't know, 60 or 70 that were consistent across 3 methods or more. So there's some on that, but from a lot of like even outside the bipolar and like psychiatric genetic space, we see a lot of inconsistencies across methods, like for fine mapping, whether it's TWAS or fine mapping and so forth. It's definitely an area that I think needs to be worked on.
I was just going to
Okay,
jump
good.
in and add, yeah, that point, like at the very end of my PhD, when I was finishing analyzing all my long read sequencing, like the current databases that we have, we found so much variation in our samples that was just not present in existing databases. And I think Cal really beautifully answered it in that, like, the more we do it and the more we look at those regions, the more we'll start to find what's true variation and what's individual variation.
We've got 2 more questions, but I'm not sure if we're able to continue because we're beyond the hour. Ailey Mara can give us advice on that. Do we have time for 2 more questions, or do we have to close?
We can go on.
We can finish them? Okay, great. So, Cal, any plans for the team to functionally test protein-protein binding for the example you showed to validate functional consequence variants? That's a really good question. Yeah.
Yes, definitely. So we're pulling out the missense variants in that region that we think are important. And then we're going to functionally model those in cell lines and see how they interact with like how DOP1A and ATP9A interact.
Very cool.
The next one, you go on.
No, you do it.
For single nuclear variance level analysis, currently whole exome sequencing for rare variant studies and SNP analysis for GWAS is cheaper compared to BGE. Do you see any future trends to shift into BGE?
I think at least a lot of our collaborators have shifted towards BGE. From a cost perspective, I think it makes more sense. And we've also made several pipelines that are just publicly available, including a BG toolkit to try to make it as easy as possible for people to use the technology. And then the Broad has released it as both a regular product for research and also a clinical BGE product for people who want to do more clinical research as well. So I do think there will be potentially more adoption unless whole genome sequencing, the costs of it and also the storage of it really scales down much lower.
My understanding, Cal, as well, is that BGE gives you a greater flexibility for investigating different population backgrounds compared to the genotype array, as the question was asking about, that the array data can be very Eurocentric and Eurobiased, whereas the BGE is it doesn't have that same inbuilt bias, which is, to me, is an amazing thing. Yeah,
that's definitely the main benefit of using the BGE over a microarray. We've seen this for we've benchmarked this with samples from Africa, and it, like, microarrays just perform awful for a lot of the samples, and whereas we use capturing all the common variation.
And the cost.
And the cost, and then, but in parallel with that, for global projects, also having the telomere, the long read sequencing, meaning we have better representation, we have telomere to telomere representation of the genome, so better accessibility, but also we're looking at more population specific haplotypes. So I think this the blended genome exome is a wonderful tool, but I think it was Natasha had said about blended short read and long read sequencing. I think we really have to recognize the value of all the different methods that have been presented today and how they actually are probably going to be used together to improve our ability to identify rare variants across the spectrum of variant types in psychiatric genetics research. It's been a brilliant session. I'm really grateful.
Yeah, so having said that brilliant conclusion from Niamh that we actually need the three of the methods, I would like to thank you again for participating. Great presentations. This was really a wonderful session. And see you in the next session and hopefully all in Glasgow. So thank you again and bye. Oh, just remember this will be posted in the YouTube channel so you can again see it because it is really worthwhile also for your students.